Hey, you guys, I know that you're doing very well, and we are going to learn the next part in this chapter. We have learned so far the structural basic information, we have learned unicellular, multicellular, cell membrane, the basic structure. Now we are going to learn the organelles of the cell. I'm going to share my screen and let's start it. Okay, so here you go. Let's start. So basically, guys, let's start it from the list that I'm going to cover. So basically, we are going to learn all the organelles of the cell. We're learning about mitochondria, ribosome, and endoplasmic reticulum. Did I select the wrong pen color? Okay, yeah, I suppose. So basically, we'll be learning all these organelles. So let's not waste the time and let's start it there. So the first one that we are going to cover is ribosome. No, I did not tell you. I have told you this. Ribosomes are actually formed up into the nucleus. So inside the cell, there is the nucleus, and this is the place where ribosome is formed. And then through the nuclear pore, these just come out. I'm going to explain the structure of nucleus also, and the pores I'm going to show you. But for the time being, ribosomes, they are the actively synthesizing cells. They are active in the present in the liver cell, pancreatic cell, endocrine cell, lymphocytes, and the meristematic cells. They are actually the protein, not the factory, but they are the location where protein is formed. Ribosome, the term ribosome, it was given by a person named Pallid, and this is the reason that these are also known as Pallid particles. Actually, ribosome is acting as a location. Like ribosome, it basically consists of two subunits. One is the large subunit and one is the small subunit. These subunits, they are normally present like separated into the matrix. But when a protein is needed, they act as a machinery, and then these subunits come together and they start forming the protein. Are you getting my point? Now, let me tell you one thing, guys. There is a difference between prokaryote and eukaryotic cell at the ribosome level also. Prokaryotic used to have the 70s type of ribosome, and eukaryotic used to have 80s type of ribosome. With their subunits here, 50s and 30s, large and small subunit, and here this is 60s and 40s. And now you people might be wondering what this thing is. This S is the Sewardberg unit. Actually, this is the sedimentation coefficient. Sewardberg unit. You must have heard the centrifugation in physics as well. Centrifugation is when something is rotating in the machine at a very high speed. So this is like a sedimentation coefficient in that centrifugation. That at which speed they are settling down in a small tube kind of thing they take. So this is because ribosome is made up of protein, and this is having its own molecular weight. So obviously, when we will be moving it at a very high rate, it will settle down at a particular, you may say, value of that constant. That is what this S is, Sewardberg unit or sedimentation coefficient. And on the basis of that thing, we have just calculated that prokaryote used to have 70s and eukaryote used to have 80s type of ribosome. Now you have to remember this. There are two subunits which come together only when protein is needed. That's it. That's all about the ribosome. Now we are going to learn the next thing. Okay, if a question come in your paper that what is the function, it usually come as a 2 marks question. What is the function? You will be saying it helps in protein formation and next thing. Maximum thing in our body is made up of protein, enzymes, hormones, different molecules, hemoglobin, cell membrane, cell wall. Wherever protein is needed, there you can write the function. If it is a 2 mark question, you simply write protein formation, and then it helps in formation of hormones, enzyme, all those things because they have protein. And then if a question is a 4 or 5, then you can just talk about all the parts of the body. Our muscles are made up of protein, bones are made up of protein, cell membrane, cell wall, everything. You can just mention all the points. Okay, let's move forward now. Now the next thing. Okay, there's a video also what are ribosome. Actually, I have added it on the OneNote that I'm going to show you. You might be wanting to see it. Let me show you then. Where's my OneNote? There it is. Actually, I have explained you everything on the ribosome. If you see the video, now you will see the same information. So what they are explaining that ribosomes is formed inside the nucleus. I'm going to make it very fast. So this average mammalian cell has 10 million ribosomes. That's fun fact, made up of RNA and protein. And function is read RNA and synthesis protein. Actually, you must have heard about DNA and RNA. These are the chemicals present in a cell and they have their own function. We used to say that DNA controls all information in us. Actually, it do it with the help of RNA. DNA is like giving information to the RNA and then RNA is going to do the work. Like that's how it happened. And that RNA is taking help of the ribosome to do it. So in simple words, I'm telling you, you don't have to think like, oh my God, this is something I don't know or something like that. So these are the two subunit, large and small subunit into the RNA. Both subunit consist of protein and RNA, and RNA in ribosome is called ribosomal RNA or rRNA. I don't think too much information is needed on this, but yeah, the next thing they're talking about, this is the place nucleolus. In the nucleus, there is a nucleolus. That's a place where ribosomes are found. So here free floating ribosome come out. Yeah, I was telling you now, I'm going to show you a picture of nucleus. So this is the nucleus and these brown thing or whatever the color B is, these are the nuclear pore. Whenever ribosomes are found inside the nucleolus, they come out through this. Okay. So this is like that way. Yeah, that thing is, yeah, ribosome formation, that what they are telling. In translate, you don't have to learn that. Actually, since you have seen this information, I would tell you because you might be thinking what ma'am has left not to tell. The process of protein formation is known as translation. That word they have used. That's it. This much information is enough for you. Let's come back to the presentation and let us discuss about the next point. The next one is mitochondria. From the bachpan, from the childhood, we know only that thing. They are the power plant or the powerhouse of the cell. Now let's defold the story that why they are called like this. Why do we call them the powerhouse? Okay, I did not anything about the mitochondria. I'm so sorry, guys. I will show you. I'm going to draw it actually. Mitochondria. Mitochondria, they are the powerhouse of the cell. Mitochondria. I'm going to give you basic information. This is the place where a reaction happens, which is known as cellular respiration. Guys, give me a second because what I need is I need a blank space and I don't have it. So I'm going to escape. Hold on for a second. And keep all the content. And then I'm going to add one slide in it. New slide. Title and body. I'm gonna make, I have to share my screen again, I suppose, no? Cannot see it, actually it stopped. That's totally fine. Let me show you the PowerPoint L4 here. Yeah, I suppose now you can see my screen, now. Hold on for a second. I'm stopping the screen share, okay? Okay, I will do the screen share once I share my screen. Now you can see it now. Forget the system issue. I have it on now. Sorry for taking a few seconds. Okay, so now guys, let me continue at this, that into the mitochondria, actually, this is the place where cellular respiration happen. If I say that this is helping in the formation of energy, I have told you this earlier also, whenever we talk about energy, it means ATP, adenosine triphosphate. So mitochondria is the place where that ATP is formed. And this ATP, it used to formed by a reaction, which is known as cellular respiration. Like suppose this is a cell and inside the cell, this is the mitochondria. We used to say that we need, breathing and eating is the most important process that we do. While we eat, ultimately our food is converted into the glucose. Through the blood, it enters into all the cells. So this glucose comes into the mitochondria of all the cells. And when we breathe oxygen, through the air, we are inhaling oxygen. So this oxygen through the blood, again, it is transported to all over the body cells. And inside the mitochondria of every cell, glucose and oxygen react. And after reaction, they form ATP. Plus the waste product, which includes CO2 and rest you will get to know. And that is the thing. So eating and breathing both are important. And this reaction is a cellular respiration where ATP is formed, where glucose and oxygen molecule react inside the mitochondria. Now, I will give you information about the structure of the mitochondria and where the story happens. Talking about the structure of mitochondria. Mitochondria, it is a double membranous structure. It used to have outer and inner membrane. Outer membrane is plain and inner membrane is folded. The good thing is I have a video. I forgot to add the picture here, but that's fine. So inner membrane is folded and outer is plain. This is inner membrane. And the space between the two of them is known as perimitochondrial space or intermitochondrial. Okay, I'm just writing it. Perimitochondrial space. Now, what's inside the mitochondria? Inside the mitochondria, these, what do we call, many things are present. Now, what things are present? Let me tell you that. Inside the mitochondria, there is ribosome. And then internally, there are all ribosome. Yes, mitochondria also have their own ribosome. Mitochondria has their own DNA also. The reason that they have DNA and ribosome, of them, they are having their own protein, own all mechanism. That is why mitochondria is also known as semi-autonomous organelle. Autonomous means independent and semi means half. So semi-autonomous organelle. It has its own ribosome. It has its own genome, genetic material. Okay. So now one thing, inside this membrane, not inside, I would say onto the inner mitochondrial membrane. Let me just change the pen color and highlight it. I need a highlighter. So this is the place onto the inner membrane where ATP synthesis happen. So basically at this place, okay. I need a pen. Yeah. So basically what happened onto the inner mitochondrial membrane, there is an enzyme present, which is known as ATP synthase enzyme. ATP synthase enzyme, ASE. So this ATP synthase, this is present all through. That's why this membrane is folded inside. More the foldings, more of the enzyme is present and more of ATP is formed here. So many of the ATP synthase are present and these are the one which are forming ATP. These are the one where the glucose, oxygen, they are reacting together. They are showing cellular respiration and they are forming ATP. Remember. Now one more thing. ATP synthase, this is also known as F-note F1 complex. Actually, I suppose there's a problem in writing in this area of the presentation. So I'm going to write it here that F-note F1 particle. ATP, this is also known as F1, F-note F1 particle. And these are actually the enzyme which form ATP. And forget all the cuts that I'm making. I'm not intentionally doing it. That's the problem with my system only. It's fine. I'll get it rectified in your next video. Now let's continue. I needed a blank space and I have used it all in the last picture. Okay, so in this blank presentation, in this blank part, I'm going to stop the screen share, guys. I'm going to show you the diagram actually. Mitochondrial diagram I need. So you just need to give me a second. I will show you the diagram of mitochondria because that's much needed. Okay, I have added it. And let me start the screen share again. You can see the diagram, right? Yeah, so that's I was talking about that it is having the outer membrane. This is having the outer membrane and this is having the inner membrane, and in between the space which is known as perimitochondrial space. They have not mentioned it in the diagram, but I've explained you. And one more thing, these foldings of the inner mitochondrial membrane is known as crystal. Inside they have shown the ribosome. Granules are actually the product, the storage bodies. These are present, the stored material. Can be carbohydrate, something like that. Okay, and the cytoplasmic thing, actually we call it as matrix. We do not call it as cytoplasm. This is the fluid part. Cytoplasm has like structure only, but we call it as the matrix. These are the ATP synthase particles. I have told you these are known as F node and F1. Clear? Okay. So DNA is there. I don't think any more information is required. Let me see if I have a video on mitochondria. I suppose I've added it on my OneNote. Let me just check it now. Yeah, this is there now. I will play it from the place wherever it is needed. I will not show you this woman. She's talking.
Storage system tops the chart of requirements. The work, there has
I don't have to tell you. I don't have to tell you this much information. I'll just come to the point directly.
Of course not. The enzymes in the mitochondria convert these energy molecules into usable molecules on the adenosine triphosphate. It's abbreviated as ATP. So are these ATP molecules the usable forms? That's right.
So they are this woman, she is telling that I have to just mute it up. I'll just speak by myself. So yeah, she was saying that there is ATP molecule, ATP synthesizing molecules are present and they are forming. See in the diagram, they have separated outer and inner structure. By looking at the video, I don't think is there any extra information that I've not told you. So you can check it directly here. And I'm going to come back here. I'll continue with the next point. I'm done on this. Now, guys, the next thing that we have to cover is plastids. One thing about the plastid that you have to remember, that plastids, these are the storage bodies. Either they are doing some function, they have a pigment, or they have, these are the storage bodies. They might have certain pigment or might have certain pigment only, which is covered by the membrane, and these pigments are doing something. I'll just let you know. Basically, plastids, these are present in the plant cell first of all. These are, there are three type of plastid: leucoplast, chromoplast, and chloroplast. Now remember one thing, guys. Wherever you see the word leuco, that means something is white. And wherever white, or you may say transparent, and wherever you see chromo, that means something is color. And wherever you see the word chloro, that means something is green. Okay, so these are actually depending upon the type of pigment which is present inside. Now talking about the first, leucoplast. Leucoplast is a storage body, a membrane which is having a stored material in it. This is further of three types: amyloplast, elioplast, and alluroplast. Guys, one thing that you have to remember is what things are present in it. So basically, leucoplast, they are the colorless pigment, and inside amyloplast, starch is stored. They store, where do I write? Starch. So they, on the screen, they have written the example that this is present in the potato tuber, wheat, and the rice grain. And the next one is elioplast. Now, do you know what is elioplast? Please tell me, guys. Okay, in the video, you cannot tell me, but you can just tell me when I'm not in the video, when you meet me probably in the session. Elioplast, they are the one which are storing the fatty substance. Oily, or you may say the fatty substance. So example is castor endosperm, tuberose. Arey, matlab jahan se bhi any plant is there, from wherever we are getting the oil, that thing. Alluroplast, they are wherever you listen the word allurone is a type of protein. So it's storing the protein. Example is allurone cells of maize grains. So I suppose that is clear to you. Now let's talk about the chromoplast. Don't tell me I did not add chromoplast. Okay, I have added it here. So there is a blank space. If I need it, I'll just come back there. I don't have to write anything on this. So chromoplast, I have told you, these are the colorful pigment. They are the colored plastid. The color can vary from red, orange, blue, violet, all those colors. I don't have to tell you all the colors now. So because they have the pigment in it, that can be a carotenoid pigment. Many pigments are present. Cyanin pigment, anthocyanin, and erythrocyanin. These are mainly the pigment. The blue or red pigment which is present, but other than this also carotenoids are of many types. These pigments are present. They are mainly found in the mesophyll cells. They are the double membranous organelle and two distinct. So in this, what they have explained is this mesophyll cells. They have actually explained about the chloroplast. I'll tell you this separately because I have categorized it as like a separate thing, third category. So I thought like it is did I make this presentation? Okay. I had to update according to me. Okay, so basically, I'm going to come back to the chromoplast only. So there are mainly these chromoplasts. These are mainly present in flower and fruits. Wherever you see the colorful flowers and colorful fruits, they used to have this pigment. Fair enough. So they provide coloration to organ mainly in flower and fruit also to attract the pollinators. Xanthophylls, carotenoids, and carotenes. Yeah, these are all type of the pigment which is under the chromoplast, and they are actually of beautiful color and beautiful type. And that's it about the chromoplast. Now the next information I have to provide you is about the chloroplast. I see I don't have space for the chloroplast. I'll come back here. I'm going to talk about chloroplast on this screen, guys. As I was telling it on the next screen, not the last one here, that they are present in the mesophyll cell. First of all, chloroplast helps in photosynthesis. You know that, right? Into the leaves, maximum photosynthesis happen in the leaf, but it happens in all the green part of the plant, but maximum in this. Reason being, maximum chloroplast, maximum chlorophyll in the chloroplast is present in the leaf. Okay. So into the leaf, there is a special type of cell which is known as mesophyll cell. This mesophyll cell is the place where they are present, and mesophyll cell is like a normal cell, but it has a number of chloroplast in it. That's a modified version of it. Clear? So in it, chloroplast is present. Now, why green color? You know that it's chlorophyll. Now, the important thing about the chloroplast that we have to learn. One thing which is common as that of mitochondria, I told you, they have their own ribosome. They have their own DNA. And that's the reason they are also known as semi-autonomous organelles. Now, guys, remember one thing. There is a theory. I'll suggest you can search about it, which is endosymbiotic theory. Endosymbiotic theory says, it's additional information that I'm providing. It says that mitochondria, chloroplast, these used to be like the separate organelle. मतलब जब दुनिया बनने लगी थी, cells मतलब living organism. So when the time they were getting formed, chloroplast, mitochondria, and all the organelles, they were like separate into the environment. And then gradually they start coming together, and then they were covered by the membrane. At that time, chloroplast and mitochondria, that's why they're semi-autonomous. They were functioning separately, and they had their own protein on they are having their own DNA. They were working separately, but then eventually they got together. Is that clear? That's the reason. So ribosome and RNA. Now talking about the structure of chloroplast. Chloroplast also used to have double membrane. I'm bad in drawing, I suppose. Double membrane, outer and inner. Now one thing that you have to remember. Yeah, perimitochondrial space was there, and here chloroplast, inner chloroplast piece, you may say, it's just in between the two membranes. Now talking about what is there in the chloroplast. Chloroplast used to have mainly two parts, grana and stroma. Right? These are the two important parts. Grana look like a stack of coin, like a pile of coin, 1 over 1 over 1, like this. Number of time it is present, I'm just drawing it like the 2 time. Now these grana, these are attached with each other with the help of a channel. We call it as stroma lamellae. Actually, the stroma lamellae, these are also known as Fred channels. Fred channels. These are the ones which are connecting. Now, one thing. Grana look like a stack of coin, and the one coin of the grana is known as thylakoid. A number of thylakoid together makes the grana. And the matrix part is known as stroma. Clear? This is the structure of chloroplast. Now add on information. If you have ever learned about photosynthesis, photosynthesis is the reaction which helps in making food for the plant. Photosynthesis used to happen in 2 steps, light reaction and dark reaction. Light reaction and dark reaction. Now, into the light reaction, oxygen is formed. Remember this. Into the light reaction, energy is formed. Now, listen to me carefully. This energy from the light reaction go into the dark reaction, and there this energy helps in making product of the photosynthesis. Glucose, the carbohydrate, glucose, starch, whatsoever, that's a carbohydrate. Carbohydrate is a category under which all the glucose and all those things come. Chloroplast is very important topic. It can come as a 5 marks question, and many students get confused at this. So this can be asked, and I hope you're listening to me carefully. So light reaction and dark reaction. And point is that into the dark reaction here, glucose is formed and CO2 is fixed. CO2, this is fixed. There is some problem with the PowerPoint thing. I'm going to get the issue resolved later, but you understand this point. Light reaction and dark reaction. Now, one thing that you have to remember, light reaction used to happen in grana and dark reaction used to happen in stroma. I repeat myself, light reaction, here oxygen is formed, go into the air. Energy is formed in the form of ATP. Then ATP go into the dark reaction. Means this whole story is happening in the grana. Then it moved to the stroma, the ATP. And into the stroma, the dark reaction happened where CO2 comes from the air fixed from the glucose as a product. I mean the carbohydrate product, which is later on stored as a starch. That's all about the chloroplast. I hope that is clear to you. If you have an issue again, you can post it on the forum. Ask the expert option on the website. You just go there. You will just see. Now, if a question come in your paper, what is the function of chloroplast? You can clearly write it. This is the one which is converting solar energy into the chemical energy. And if a question come on the plastid, you can just mix all the points about the leucoplast, chromoplast, present in fruits and flower, attracting the pollinator, leucoplast, storage thing, chloroplast, helping photosynthesis. Just mix all the points. I'm done on this. Now let's move forward. The next thing, okay, so sorry, I forgot to show you the diagram itself. That's chloroplast. Outer membrane, yeah, that's inner membrane space. Outer and inner membrane is there. This is thylakoid membrane. So thylakoid also, it has its own membrane. So inside the thylakoid space is there. Like suppose this is a thylakoid. So internal space is like the cavity kind of thing, which is known as internal lumen. Lumen means the internal. Like suppose this is the pen. Inside the pen, the cavity is known as lumen. So lumen is like a generic word. It's not a terminology. So thylakoid space and yeah, thylakoid and stroma and ribosome and chloroplast. I suppose that is clear to you. Now let's move forward. Oh, sorry. I forgot to show you the video on the chloroplast. I suppose I had it. Let me just see it on my OneNote, I suppose. No, I have it on nucleus. Okay. I'll come back here once I talk about the nucleus, because the next point is nucleus only. Let me see how much information I have on the nucleus. I'll just come back here. So basically, guys, nucleus, this is the controlling body of the cell. This is the mother of the house. Iski ghar pe chalti hai, I suppose so. So nucleus is the control body. This is the one which is having genetic information. Nucleus is having a number of parts. I'm going to show you a basic diagram here. Like suppose this is a nucleus. Guys, I suggest you one thing. There used to be two type of diagram. One is known as 2D and 3D diagram, right? Like the picture that you see right now, this is a 3D diagram. 2D diagram is the one that I'm drawing. 3D is three-dimensional one. Once you're doing it in the paper now, you should go for the 2D diagram. That's easier to make, easier to understand. And also student used to ask about the DNA, the chromosome, all the story I'm going to explain you. Inside the nucleus, nucleolus is present. And then onto the nucleus, there are nuclear pores. Actually, while the time you make nucleus now, you should make it like that way. And let me see if undo key work on this. I've never tried, but it's not working. Okay. So basically, I'm going to draw it one more diagram here. While the time you make nucleus now, you better make it like that. When nucleus used to have double membranous organelle and used to have pores on it. So the one that I'm trying to draw is pores and the double membranous organelle. You should make it in a circle. Okay, I have a very beautiful drawing, but it's on the paper, not here. I'm using the digital pen and tab, so I'm trying my best. So you make it like better that way. Inside the nucleus, the nucleolus is present. So this is nuclear envelope, or you may say nuclear membrane, and it is a double membranous. So two times I have drawn. This is the double membranous. So this is nuclear envelope. And these are the nuclear pore. You should write nuclear with it. I'm not writing. I'm just saving my time. Okay, this is nucleolus. Then inside the nucleus, there is, I should draw it with a different color. Inside the nucleus, then there are genetic material present inside, chromatin. And then inside a number of ribosomes is formed. I repeat myself, ribosomes are formed inside nucleus, and after getting formed, they come out. And then they come out simply, they join wherever they want to be. Now, where they join, I'm going to explain you that as well. Now, one thing that people get confused with, DNA, chromosome, and chromatin, how they are related. I think I have to press and escape and come back. Probably my pen, it would start working fine. Chromatin, how they are related. Let me explain you. Chromosome is made up of DNA. Chromosome is nothing but it is highly coiled DNA itself. Extremely coiled DNA that appears to be like a chromosome like this. Actually, there are stages. First of all, a chromosome is like double membranous, double coiled helical structure, double stranded. This is the one simple. It keeps on getting complex and complex. It's forming complex, 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 complex. It appears to be like a thread-like structure. The thread-like structure stage is known as chromatin. Now, this chromatin then keeps on getting complex and then ultimately it takes shape like a chromosome. So chromosome is nothing but it is extremely, extremely coiled DNA. Fine. Now you will remember this, right? I was about to say one thing on this. Yeah, I was saying that chromosome, normally inside the cell, chromatin material is always present. Like this is not the DNA I'm drawing, this is the chromatin I'm drawing. Normally inside the cell, this is always chromatin which is present. But when it comes times of the cell division, listen to me carefully, when there's a time of cell division, only then it takes shape like a chromosome. Clear? Good. And one more thing I was saying about the gene. So many people used to get confused about the gene also. So gene are actually the segment used to be present on the chromosome. Like gene of color, gene of height, gene of whatever character is there. So this is present onto the chromosome. So gene is made up of the DNA, but these are the segments, unit of inheritance. I suppose I'm done with the nucleus, but I have a video on this. I'll show you, but first see this. Yeah, so the cytoplasm matrix thing is known as nucleoplasm. Let me change the pen color. So nucleoplasm is the cytoplasm thing, and chromatin, I have explained you, perinuclear spaces between the two membrane, and nuclear pores, nucleoplasm, nucleoplasm is written 2 times. Edit, ribosome. Okay, so there you see the diagram, guys. This is the 3D diagram. 3D diagram is easier to understand, but for drawing, I suppose you should go for the 2D one. So this is the nucleolus at the center. Then there are condensed chromatin. This is nuclear envelope. These are the nuclear pores and nucleus. Now remember one thing, guys. Nucleus is always covered by endoplasmic reticulum externally. Endoplasmic reticulum, everyone knows about it. Ribosome formed inside the nucleolus immediately go through the pores and join endoplasmic reticulum. There it is present. Okay? People also get confused at this. You need to remember this. Okay, so many students actually ask me this question that what is the story of ribosome? So it is found inside the nucleolus. Now, guys, I don't think an explanation on is needed on this. Okay, while the time I write, animation stops. I did not know that. You can see. Okay, I'm going to talk about the endoplasmic reticulum, but I showed you in a video. So let me show you that video. The new share of my screen, coming back to your notes. And yeah, there is the video. I'm going to forward it because I suppose I have explained you everything about it. I'm going to put it on mute. The same energy cellular organ. Okay, so in this video, what they have explained is the chromatin fiber, like all the thing I was explaining now. See, inside the nucleus, they are chromatin fiber, which are important to us. They're the most important component. Okay. I think I should be silent and just show you this video. एक सेकेंड ना मैं थोड़ा इसका साइज इंक्रीज कर लेती हूँ. Okay, so I was just increasing the size. Now you see the video. I don't think I have to speak. So when the cell has to divide, these long thread coil themselves to form the sophisticated structure that we call it as the chromosome. So in this video, actually, they are all showing all the stages that from the DNA, how come chromosome is formed. And I have explained you this thing. It's just that you are watching it so that you can just see these are all the genes and DNA. This is, yeah, double-stranded DNA, which is forming it up. I'm coming back to the presentation. You have seen it. Thank you. So now, guys, let's talk about endoplasmic reticulum. See, I was telling you one thing, that this is the nucleus, and outside the nucleus, there is always, what a worst diagram I have made. There is always the coiled structure present like this. It appears to be like the coils, not the coils, but I will say folded sac-like structure, 1/1 like that way. So immediately when ribosome come, they join it. And this is endoplasmic reticulum. Endoplasmic reticulum is actually of two types, rough endoplasmic reticulum and smooth endoplasmic reticulum. Now, wherever ribosome is present, that area is known as rough endoplasmic reticulum, and where it is not present, that is smooth. Now, we will be watching their structure and nothing. Let's see the diagram. So here you see, I suppose you're able to see this picture. Here you see this is a nucleus, and onto the nucleus then endoplasmic reticulum is present. And okay, I suppose you might not be able to watch this area, but that's totally fine. My main point is show you endoplasmic reticulum. This is rough and this is smooth. If you look very carefully at it, rough endoplasmic used to be looking like these folded sac-like structure, but smooth endoplasmic reticulum looking like this. Actually, these are the tubular structure. And endoplasmic reticulum is having all the folded structure. Now, guys, these are actually known as cisternae. I suppose you know that. These are basic information. There are three things into the endoplasmic reticulum. One is known as cisternae. Then there is tubules. And then there is vesicles. Okay, so cisternae look like this, tubule look like this, and vesicles are small vesicles. Okay, so in the diagram that I was showing you, rough endoplasmic reticulum mainly consists of the cisternae and few tubules only. But then smooth mainly consists of tubule, and smooth doesn't have all the ribosomes over it. So on the diagram, on the screen, you can see the differences. It possesses ribosome attached to the membrane. It doesn't have ribosome on its membrane. This is formed of cisternae and few tubules. This is formed of vesicles and tubules. Remember this. Now, one thing that you have to remember, the function. I have told you wherever ribosome is present, this is forming the protein. Yeah, they're the protein factories. So here endoplasmic reticulum, they are the one rough is having the ribosome. So it is helping in the formation of protein, and whatever protein is forming. It is forming enzyme, hormone, whatsoever. Now, this is synthesizing smooth. I'm talking about this is synthesizing lipids mainly, steroids, glycogen. If a question come in your paper, you can simply write it. See, if a question come in a paper, you need to write function of endoplasmic reticulum. You can write it as a mixed point. This is a tip for you guys. While the time you write the question on the organelle, you should write it as the structure, function, structure, function. So into the function, you mix all the point. Smooth endoplasmic reticulum, rough endoplasmic reticulum, mix all the point, and then you can also say they help in cell membrane formation, because cell membrane is made up of protein and lipid. The process of cell membrane formation is known as membrane biogenesis. I repeat, membrane biogenesis. Genesis means synthesis. So it helps in that. It also helps in the glycogen lipid. Guys, if I talk about one thing, male and female used to have the hormones. We call it as sex hormones, testosterone, progesterone, estrogen. They all are made up of lipids. You can say here, if you're talking about the point, that it helps in forming the hormones, the sex hormone. They are made up of lipids. Remember it, okay? So I suppose I have told you a lot of information about endoplasmic reticulum. Again, if you have any doubt, just put it on the forum. Clear, guys? Ask the expert thing. I'm going to move forward. Let's move. Okay, now the next thing that you have to cover. Organelles of the cell may, yeah, Golgi body is also known as Golgi complex or Golgi apparatus, and dictyosome is actually, we call it in case of plant cell. In case of animal cell, Golgi apparatus is there. In plant cell, we call it as dictyosome, almost same structure, doing the same function. Okay, and acha, sorry, last May I did not mention about the vesicles now. In the diagram, upper wale they have not explained the vesicle. Here they've explained the vesicle. These are, see, on the Golgi apparatus, these vesicles are. Okay. वो यहाँ से याद आ गया मुझे, that thing reminded me from there. Sorry for speaking Hindi, but I'm repeating in English and I will not do it in the next parts. Okay, so they are the one which are producing lysosome. One thing. Golgi bodies, they are actually the place where packaging happens. Product is ready in endoplasmic reticulum, protein, lipid, whatsoever. Now that protein, lipid is not functioning. It's very raw structure. For modification, for packaging properly, it comes in the endoplasmic, sorry, it comes in the Golgi apparatus. There it comes, proper shaping is done. The shape it should have, the package it should have, then it is modified here. So this is a packaging place. Here modification happen of any structure, and you may also call it as the proofreading of the structure. If any error is there, this is rectified. So into the Golgi apparatus, this mainly consists of three things: cisternae and the vesicles. These are actually the two things. Now, one thing that you have to remember: cis phase and the trans phase. Cis phase is like the immature phase. Here, like suppose this is endoplasmic reticulum. From the endoplasmic reticulum, a protein which has been formed, which is packed in the vesicle. Vesicle is like a bubble kind of thing which is taking the molecule and going it toward the Golgi apparatus. The protein or lipid whatsoever has been formed in the vesicle, it will come toward the cis phase, which is the immature phase. And then inside modification will happen. And through the trans phase, it will take the exit in the form of secretory vesicle. It might be having protein, lipid in the modified functioning form now. So trans phase is the mature phase. So if it is saying that they produce lysosome, lysosome is formed, is actually the structure which has a number of enzymes in it. And those enzymes are made up of proteins. Getting my point? So basically, lysosomes are also formed because into the vesicle, then number of enzymes will be present, and we call it as lysosome. Something is wrong with my tongue. So I suppose that's clear. It is secreting various enzyme, hormone, cell wall material, protein, and lipid, everything that is made up of that you have to remember. Peroxisome, lysosome, that's another vesicle kind of thing that is formed here. And I suppose Golgi apparatus is very much clear to you. Actually, in the last diagram, there, yeah, see, this is endoplasmic reticulum. Here, transport vesicle is coming. And I'm so sorry, guys. Okay, so this is the transport vessel. From here, it is coming inside, and then it is, after modification, this is taking an exit. This is endoplasmic reticulum, and this is Golgi apparatus. From there, it is coming, and this is taking the exit. You see. Clear? Why it is going next by itself? I didn't do it. It's telling me to move forward. You understood it well. Okay, let's talk about the vacuole. How many space I have? Okay. So now, guys, the next thing that we have to talk about is the vacuole. See, vacuole is a place which you say is the extra place. Extra place as in that thing is not required or required in the cell. Like suppose in a cell, yeah. Suppose, first of all, vacuole is present mainly in the plant cell. This is rarely present in the animal cell. That too in the small unicellular animal cell, not in us. Suppose amoeba. I mean, in many of the protozoans. Amoeba is one example, not in higher organisms. Now, vacuole is larger in plant cell and not that much larger in animal cell. Talking about the plant cell or any cell, vacuole is like an extra space. If any useful material which is not needed, present in vacuole. If it is needed, it will come out. If it is not, it will go back. Even waste material is also stored there. So these are membrane bound fluid filled cavities, and the membrane of the vacuole is known as tonoplast. Remember this. Okay, so the vacuole, with they're having the fluid, that fluid inside is known as cell sap. Now talking about the types of vacuole, let me see if on the next screen I have written it. No. Vacuole is mainly of three types. One is known as sap vacuole. Second is known as food vacuole. Sorry, it's UOLE. And the third one is known as contractile vacuole. Again, sorry for the writing, as I'm trying to write on the best writing, but it's some internal system problem. I'll take the IT help. Okay, so sap vacuole. Sap vacuole is mainly present in the plant cell. It used to store salt, sugar. Listen to me carefully. If the pen is not working, you should listen to me carefully. And some proteins. And some proteins. Let me change the pen color. And listen to me carefully again. Sap vacuole helps in maintaining turgidity and rigidity of the cell. Turgidity means a cell is firm. A cell is very firm and having a proper shape. So it helps in maintaining that thing. It also helps in maintaining osmotic pressure of the cell. Osmotic pressure means the pressure which is because of the fluid and liquid. The osmotic pressure that is maintained. Right? Now talking about the second type, which is the food vacuole. Food vacuole is actually, we call it in amoeba. Actually, food vacuole is like a temporary vacuole. In amoeba, in paramecium, food enter inside the cell, and then inside the cell, food is covered by membrane, tonoplastic cell. We call it as the food vacuole. So it is present in the unicellular organism. Guys, I don't know whether you have heard or not, but the way these unicellular organisms eat the food, that is known as phagocytosis. So inside the phagocytosis, when food is moving inside, we call it as the food vacuole, which later on is digested with the help of enzymes. Now, which enzyme? That is lysosome. I'm going to explain you that later, but that is the food vacuole. Clear? Achha, one thing. A food vacuole is known as phagosome, phagolysosome. You simply call it as hold on. Phagosome, phagolysosome. No, phagolysosome will form later. Yeah, you just forget it. I'll explain you that later. Basically, you just remember it as a food vacuole only. Now, the third one is contractile vacuole. See, contractile vacuole, this is the one which is collecting liquid from the cell. Inside, you may say unicellular organism like amoeba, it is present. So inside the cell, what is going to do? It is going to take all the waste material from the cell and it is going to swell up. And then after swelling up, it will move toward the membrane and burst and release everything out there. You're getting my point? By doing this way, this is also help in maintaining osmotic regulation. It is removing all the waste product. We call it as osmoregulation. And second, it is also helps in excretion through this way. Excretion and osmoregulation. That's all about vacuole. I suppose that is clear to you. Now, if a question come in your paper where you have to talk about the function of vacuole, guys, you can write separately. In plant cell, this is helping in providing turgidity and rigidity to the cell. It is acting as a storage house for the pigments and the waste product. It is storing a number of minerals and salt. It is also helping maintaining osmotic potential, osmotic regulation. Talking about the animals, in animals, you can talk about the food vacuole and the contractile vacuole. I don't have to give you further information on this. I suppose that is clear to you. Now let's move forward, guys. Here you see. Acha, sorry, I forgot to show you this picture. This is plant vacuole. Appear to be like only bubble, salt, mineral, everything is present. And that is the reason vacuole is like a big bubble-like structure. And that's the reason that cell nucleus is present at the periphery. Clear? Now let's move forward. The last, I suppose that's the last one. Huh? Yeah, that's the last one. Organelles of the cell. The last one is lysosome. I have told you this already, that lysosome is formed from the Golgi apparatus. Like suppose food vacuole, lysosome, digestive vacuole. Yeah, they have shown phagocytosis only here. Lysosome. Okay, these are the lysosome. You know what I was trying to show you? I was trying to show you how lysosomes are formed. The picture I have not added here, but you can find this picture here. These are actually secretory vesicle now. From the lysosomes are formed from the Golgi apparatus like this only. So this is a lysosome. And the picture that I've shown you here, this is lysosome coming out from that place. And what happened inside the lysosome, this diagram is showing that. This diagram. So lysosome is like a membranous structure which has a number of enzymes in it. Enzymes are made up of protein. So this is intracellular digestive enzyme. Lysosomes help in eating out. It removes all the waste product. Like suppose a cell is damaged. Obviously, after getting damaged, it is going to release some toxic substance, right? So that is eaten up by the lysosome. Like suppose there is a natural phenomena also which is known as aging. We show aging, but a cell also show aging. After aging a cell, I am so sorry guys, I had to just keep my phone on silent. I'll take, I just got, let me just put it on silent. Okay, I'm just getting to know a few points only during the time while I'm teaching you. Done. Okay, so basically now guys, the point that I was saying that suppose a normal aging process is happening, inside aging also a cell gradually going to die and just disintegrate. That thing is also eaten up by the lysosome, and that is the function that you have to remember. So this is involved in intracellular digestion activities has, yeah, so basically it used to have lytic enzyme. Lytic means lysis, cell burst, everything is out. So it contains hydrolytic enzyme, and so that's why it is known as lysosome. They are the suicidal bag. Everyone knows that. I don't have to tell you this. They're absent from the prokaryote, but they are present in all eukaryotic cells. In this diagram, that is, they have shown that, see, acha, in this diagram, they have shown this. See, this is a cell. I did not see the diagram very carefully, but this is endoplasmic reticulum. From the endoplasmic reticulum, material come into the Golgi apparatus, and from the Golgi apparatus, lysosome is formed in the form of vesicle. So now here food is coming, food vacuole. Food particle, endocytosis happen. Endocytosis means when something is entering inside. A phagocytosis, I told you now. Phagocytosis is of two types, exocytosis and endo. Endo means when something is coming inside. Exo means when something is going outside. So endocytosis is coming, happening, and food vacuole is happening. This is food vacuole. I explained you the type of vacuole. Then this food vacuole would react with the lysosome. Lysosome has all the lytic enzymes. It is going to eat upon the food. And all the waste material, digestive vacuole, is going to be released outside. Exocytosis and elimination of the waste product. And useful material stays inside. This is how lysosome is helping you. Now, guys, if a question comes, I suppose you can clearly write on the lysosome, on the function, on the structure. I'm done on this. Let's move forward. These are some of the questions that you see. Why lysosomes? They are known as suicide bag. They when there is a damage to the cell, when revival is not possible, lysosome will burst and enzyme can just eat up everything. That's the reason. Where are proteins synthesized inside the cells? I don't have to give you answer. These are the questions that I should ask you. And I'm telling you answer itself. See how lenient I am. Thank you. I'm done on this. I'm going to stop the screen share now. Okay, guys, so today we have learned all the organelles of the cell, and I suppose everything is clear to you. Now, again, if you have any doubt, just put your question on the forum, in the discussion board in your student account, and on the public forum, like on our website, there's an option Ask Expert. You simply go up there. Is that clear? And now in the next part, there are a few important things. We have almost covered the chapter, but still there are a few important things that we are going to cover up in the next part. So I'll see you up there. Till then, keep learning what's up. Bye. Take care.